Hanski, I could probably spend almost the whole hour going through various honors and etc. I'll just quickly say that he received his D.Phil from Oxford, which is what the equivalent of a PhD, so they would probably say it's better. And he is currently a academy professor at the University of Helsinki, and he essentially is written the book, actually several books, on metapopulations, which I'm sure we will hear about today, as well as butterflies and perhaps dung beetles. So it gives me great pleasure to welcome Ilkka Hanski for his second lecture.
Thank you, Alan. Well, since yesterday's lecture, I had a wonderful dinner with some of you last night, and I have spent a very busy but very stimulating and interesting day today with some of you. So thank you to everybody with whom I've been able to interact yesterday night and today. Now, I'm coming from a department which is called the Department of Ecology and Evolutionary Biology, and one could expect that that's a place where ecologists and evolutionary biologists are interacting with each other all the time. But that's not really the case. And I believe that there are lots of other departments of ecology and evolutionary biology on this planet where also the interaction between these two groups of people are, or anyway have been, much less than what you might expect. Evolutionary biologists, of course, they are dealing all the time with genes, genotypes, and phenotypes, and the interaction between genotypes and phenotypes. Ecologist, I'm a population ecologist, so I mostly work with populations, but I'm also interested in communities, particularly dung beetle communities, and how many species, populations of many species interact in communities. Personally, I haven't done much with the ecosystems, but of course many other ecologists have, and many ecologists are interested in the interactions between these three different levels: populations, communities, ecosystems. Now, the domains of evolutionary biologists and ecologists, of course, are connected, and we all know that the process of natural selection involves the environment, the ecology, therefore, and everybody knows that, and much work, of course, thereby involves interaction, particularly between populations and phenotypes and genotypes, perhaps less so from communities and ecosystems to the level of phenotypes and genotypes. There are also well-established ideas of how the genotypes and phenotypes might influence populations, and again less so until recently, communities and ecosystems. For instance, there is the classic idea about why species geographical ranges are limited, where they are limited by the process of gene flow from the more central populations to the marginal populations, which make it more difficult for the marginal populations to adapt to the marginal conditions. So that kind of idea is well established, though empirically actually is not well established. There are also some famous conceptual models which actually very explicitly link the domain of genotypes and phenotypes to population dynamics, and perhaps one of the best one was developed by Dennis Chitty, who was working actually in Oxford a long time ago, but more recently before he died in Vancouver, and he was interested in the dynamics of small mammal populations, and particularly those small mammal populations which exhibit cyclic dynamics, like many north temperate Arctic populations do. And he was interested in the possibility that there is phenotypic and surely also genotypic variation in the population. There are more aggressive individuals, more aggressive voles, which, however, might bear the cost of being less fecund than the less aggressive voles, which would have higher fecundity. And he envisioned that in the course of the density cycle of a vole population, the frequency, the proportion of these aggressive individuals will increase when the density is high, for the simple reason that in those competitive conditions, when density is high, the aggressive ones, they benefit of being aggressive. But then they are so poor in reproduction that when the density, when the density and the frequency of the aggressive ones is very high, they just keep competing with each other, and they kind of forget to reproduce, and therefore then the population crashes, in which case then again the high fecundity, less aggressive individuals, they get an advantage. So this of course would be a beautiful example of an interaction between ecological dynamics and microevolutionary dynamics. Unfortunately, it's not true. There's no empirical support for it, but it's a good idea. In the past 10 years, and particularly the past 5 years, lots of population biologists, ecologists have been kind of become interested in the way that Chitty was interested in the kind of reciprocal interaction between ecological dynamics and evolutionary dynamics. And we now have some actual empirical examples, pretty good examples of possible interactions between these two types of dynamics. There's a long series of papers coming from Nelson Heston's lab on a predator-prey system, a lab system involving a rotifer predator and its algal prey, where there's very strong evidence of reciprocal interaction between the demographic and microevolutionary dynamics. It's a beautiful work, partly because the empirical work is matched with very nice modeling. Perhaps the sort of minor limitation in my mind is that the prey is clonal, and so actually the dynamics would be exactly the same if you would have 2 prey species and 1 predator. But nonetheless, it's an example of where the interaction between the predator and prey is influenced by the changes in the clonal frequency of this alga. There are examples, other types of studies which have analyzed long-term pedigree data for vertebrate populations, where there is very explicit demonstration of how the demographic dynamics are influenced by the phenotypic composition of the population. There are examples of plant genotype affecting the herbivore populations, even communities, even ecosystem processes, in this case from Erik Palkovak's lab and from other labs. There are interesting examples of bacterial metapopulations doing the spatial dynamics in nano-fabricated landscapes, real exciting work, and there's a number of reviews of this recent work. So that's why what I will be talking about, situations where the demographic dynamics and the microevolution dynamics are coupled, and particularly I'm interested in situations where this happens in a spatial context. And particularly I'm interested in situations where there is kind of both-way interaction, demographic dynamics influencing the microevolutionary dynamics and vice versa. Now, I want to start with an example which is much more familiar to many of you than it's to me, because I haven't actually seen this in the field. Timema walking stick insects, which is a study system which Patrick Nosil has been developing and is still, actually he's in the field right now. I have started a little kind of pilot experiment with him. Well, to me, this is a wonderful system because it's a very good example of a classic metapopulation. The stick insects, or walking stick insects, they occur in these bushes, which are habitat patches. The dispersal capacity of the insect is very limited, so you have real well-defined local populations occupying individual bushes. Here is an example of an area where within a small area of 30 by 60 meters there is this network of bushes. And the system, it shows spatial patterns which I'm not surprised by patterns which are predicted by conventional metapopulation models, namely the patch occupancy, whether a particular bush is occupied, whether it has a population of these walking stick insects, and also the population size. They are affected by the size of the patch and the connectivity of that patch to other existing populations. Now, the story is a bit more complicated, as some of you already knew before I say it now. Namely, there actually are two different host plant species also within this area, and there are two, actually a bit more, but two main color morphs of the walking stick insect. And these color morphs, they are cryptic on their respective host plant. There is a striped one, which is cryptic on the Sadenostoma species, and there's a green one, which is more cryptic on the other host plant. And there's very, very clear evidence of, in this case, ecology influencing evolution in the form of very local adaptation. So these data are coming from the 30 by 60 meter area. Patrick's students counted the different color morphs of the walking stick insect on the two plants, and you can see that there is a big difference, a highly significant difference in the expected direction. The striped one was more frequent on adenostoma, and the green one on the other host plant. So very strong evidence of ecology on evolution. At the same time, there is quite strong evidence of in the opposite direction, in the form of the genotype frequency on a particular bush affecting the local abundance of that local population. So on this graph, we have the frequency of this green morph, which is better adapted to C. nothus, going from 0 to 1, and then we have a measure of population, actually an absolute size of the population, which has been corrected by the size of the bush for the two types of plants. And what we can see is that the population on C. nothus, on this red one, is larger if the frequency of the green morphs, which is better adapted to that plant, is higher. Kind of makes sense. Whereas if the frequency of the green morph is high, but we are now on the alternative plant, then the population size is smaller. The reason for this is that there is bird predation, and the bird predation is heavier if the frequency of the wrong morph is higher. So very clear evidence, well, pretty clear evidence in this case of the genotype affecting local abundance. In this case, we don't yet know what would be then the longer term demographic consequences, but most likely if you have a smaller population, it behaves a different manner than a larger population. For instance, it may have a higher risk of extinction. So if you now want to sort of try to understand conceptually and perhaps even with models the dynamics of such a system, it's not enough to do what I've been doing for the past 20 years, namely to draw these circles and think about the effects of population sizes, isolation, and such factors on the demographic dynamics, because we now have two different types of habitat patches. We want to include that variation in the habitat type in our thinking, in our models. And particularly if you want to construct a simple model for that kind of system with 2 or more different types of habitat patches, a minimal model from my perspective would consist of describing for a finite network of n patches the state of a metapopulation inhabiting that network in terms of what is the probability that patch I in that network would be occupied at a particular point in time, with another variable which gives the mean phenotype in those patches which are occupied, taking into account that there is now variation in the phenotypic composition of the local populations. So I've been developing such a model with colleagues in Helsinki. There is no need to go into details, also for the reason that the model has been recently published. The model initially we constructed is a stochastic patch occupancy model, meaning that you don't try to model the dynamics of local populations in terms of their sizes. You just keep track of which populations, which habitat patches are occupied and which are not. But here what I'm showing is a deterministic approximation of the model, which gives you an idea of the processes that we are taking into account in the model. So the model aims to predict the time course of occupancy of all the patches in the network and the time course of the mean phenotype in those patches if they happen to be occupied. In the case of the deterministic approximation, you can solve the steady state of the model. In other words, what would be in equilibrium the probability of any one of these patches being occupied, and what is the mean phenotype when the patch is occupied. Now in the case of the occupancy probability, this depends, as you would expect, on the colonization rate and the extinction rate, whereas the mean phenotype of the occupied patches additionally depends on the strength of local selection, like bird predation in the case of the Timema example. It depends on gene flow, depending on which kinds of how much individuals are dispersing between the patches. It depends on the mean phenotype of the immigrants, which kind of individuals in terms of their phenotype are arriving to a focal patch. And it also depends on the extinction rate, which then lead to establishment of new populations, founder events, and the mean phenotype at equilibrium in the patch depends on which kind of phenotype the new populations consist of, so the founder event. Now this model, and as I said, there is no reason and no time to go into details, but if you are interested in the details, it's all in the paper. It involves several what we could call eco-evolutionary feedbacks. So there is a local possibility clearly for local maladaptation in the model, just like in the Timema example, in the walking stick example, which in the model and also in reality may lead to smaller population size. Now if the population size is smaller, that most likely increases the extinction rate in the case of these relatively small populations to start with. It also is likely to decrease the emigration rate because there simply are fewer individuals to emigrate. There of course is dispersal and gene flow, which is coupling demographic and microevolutionary dynamics, because gene flow will influence local adaptation. And there is also in the model what I call here immigrant selection, by which I mean the match or the correspondence between the immigrant phenotype and habitat quality which sets the local optimal phenotype. In other words, if you are a green walking stick insect and you are coming to a bush where you are poorly adapted, you might not like that bush, you might want to go somewhere else. And so the probability of actually settling in that patch might be influenced by this correspondence. And so it may affect gene flow and hence the evolutionary dynamics. It may also influence colonization and hence the demographic ecological dynamics. Now, in passing, I mentioned that this deterministic approximation is a very simple model, and it ignores, of course, many important processes. One simplifying assumption that we have to make here is that the variance in the phenotype is constant. So we are modeling just the mean phenotype on the assumption that the variance remains constant. And that can be quite a big assumption and could be potentially a misleading assumption. We have constructed an individual-based simulation model where the genetic variance is now modeled in a very mechanistic manner, and we have compared the prediction of the deterministic approximation with the prediction of this individual-based simulation model. And I'm showing here one example where on this panel I show the long-term probability of a particular patch in the network being occupied in the individual-based model and in the population-level model in this deterministic approximation. And you can see that there is still pretty good correspondence to obviously also scatter. And the same goes for the mean phenotype at equilibrium in the population. So I think this is sort of justification for ignoring that, assuming that the phenotypic variance is constant. Okay, so what I want to do next is to give you an example of the sort of predictions that the model makes. And I do this with an example where there are 100 habitat patches which have random spatial location, like you see here. There's variation, log normally, variation in habitat patch sizes, and in this example there are two patch types with different optimal phenotypes, the green and the black ones. Now the model is very flexible in terms of how many patch types you want to have, what is the spatial location, what is the distribution of the areas and such. But here is an example which we are now looking at. So the model predicts four, depending on the parameter values, it predicts four different patterns of adaptation, of which one is strictly local adaptation. And the local adaptation happens, as you might expect, if there is a very strong local selection, like if the bird predation would be very strong in the Timmerman example, only the very cryptic ones will survive. And also if there is a lot of genetic variance, local adaptation is more likely to proceed to a large extent. And also if the expected lifetime of local population is long, then you would expect more local adaptation for the simple reason that there's more time for local adaptation to happen. Another pattern is what I call here network adaptation, where actually in this entire network you have generalist species. In other words, the mean phenotype is about the same in all the habitat patches. Now this representation may be a bit confusing. The background color indicates what is the mean phenotype around the patch, any one of these patches, and the rest doesn't really matter. Here we have a sort of intermediate green color indicating that in all the patches you have about the same intermediate mean phenotype, and this happens if selection is not very strong. Also the demographic cost of being maladapted in this case is weak, because the same parameter that sets the strength of selection, it also then influences the cost of being maladapted. And also if the optimal phenotypes in the two patches are rather similar, the difference is small, then you are likely to have the network adaptation in the network, which again makes sense. If the two patches are rather similar, then it doesn't really make much difference if you are a little bit maladapted or not, and you kind of make compromise. On the other hand, if the selection is strong, demographic cost of maladaptation is therefore large, and now the difference in the optimal phenotypes is large, and the third parameter here refers to spatial scale of dispersal, if that is large, so the individuals are dispersing long distances, then very easily you get what we would call habitat specialization, and in this case we have a dark green color as the background, meaning that now all over the network the populations are well adapted to the dark patches, poorly adapted to the green patches, but with that set of parameters it actually pays to do that. So bear the cost of being maladapted in part of the patches, but being really well adapted in one set of patches, and this happens when the cost of being maladapted is large, so it's better to be well adapted somewhere. Now in many cases here you actually have in the model two alternative states. You may be well adapted to either the dark or green patches. On the other hand, if there would be a big difference in how many black patches there are and green patches, then of course you would be likely to adapt to the more common patch type. And finally, there's a possibility in the case of short dispersal distances that the species becomes more locally adapted, like in this example, where we have exactly the same network as in the other examples. We have strong selection, we have a large difference in the mean optimal phenotype, but now we have a short range of dispersal, and in this case the species become well adapted to one of the patches in a sub region of the network, becomes well adapted to the alternative patch type in other part of the network, and this happens in the way that by chance in this area, as you may see, there are more large black patches than light green patches, and therefore selection now favors specialization to the black patch type here and to the alternative patch type here. And because the species now has short range of dispersal, Gene4 doesn't kind of prevent that kind of regional specialization. Okay, going back to the Timoma example, we actually have fitted that model to this 30 by 60, well, it doesn't really, well, it should be a 30 by 60 meter area where these two plant types occur in a manner which this map shows. This is the observed mean phenotype, in other words, the frequency of one of the color morphs in the network, and these are the predicted mean phenotypes by the fitted model. Now this panel here shows the long-term predicted frequency of one of the color morphs. The observation of course comes from 1 year, and so you can actually produce by simulation you can produce a snapshot for one point in time of the color frequency, which we have shown here, and you can see that the model fits pretty well. Now the model shows that this coupling between the demographic dynamics and the microevolutionary dynamics has consequences for metapopulation size and viability. Here is an example where with the broken line I show the predicted size of the metapopulation in terms of incidence of particular patches being occupied. So this is the average incidence of occupancy. against the average distance between habitat patches. So we have the same network, we are just increasing and increasing the spatial scale, making the patches more and more isolated from each other. Now, without micro-evolutionary changes, so kind of turning the evolution off, we get this broken line prediction. But if you allow this coupling between demographic dynamics and the microevolutionary dynamics, we get this continuous line prediction. And so you can see that the coupling between the ecological and evolution dynamics may both decrease and increase depending on parameter values and the spatial configuration of the system, the size of the metapopulation. And in this very example, it happens because when the patches in the network are very close to each other, there's a lot of gene flow, and we end up with this generalist type of adaptation, where the mean phenotype is about the same all over the place. When the average distance between the patches increases, the amount of gene flow will decrease, and in this particular example, that leads to specialization. Instead of being a generalist, the species then evolves into this specialist type, into one of the patch types. And when you further increase the isolation, now the effective number of patches in the surrounding of any particular patch becomes smaller. So dispersal gene flow becomes more localized, and we end up with this mosaic type of specialization, where depending on the structure of the subnetwork, specialization happens into one or the other of the patch types, which increases the metapopulation size in comparison with the case without such specialization. Now, I take one example, a butterfly example, not from my own work, but from the work of Chris Thomas in England, who has worked on a butterfly called the brown argus, which is one of the species which has expanded its range due to climate change. Well, this is a kind of toy example of what might have happened in this case, but it's not a toy example in the sense that I would be making assumptions which would be contradictory with the known biology of the species. So what used to be the case towards the northern range limit of the species in central England like 30 years ago was that the butterfly occurred in two kinds of sites. Warm sites where the microclimate was warm, the red ones, and cool sites where the microclimate was cool. And it also so happens that both types of sites had their specific plant species, so different plant species. And 30 years ago, the butterfly was well adapted and preferred the plant species which occurred in the favorable warm sites. So in terms of preference between the two plant species, it was a specialist for the warm site plant. And in this example, then we run a simulation model which predicted that the metapopulation was fluctuating at this level. Now, in 30 years' time, the climate has warmed. The previously cool sites have become a bit warmer. They have become more favorable for the butterfly in terms of larval development and such. And indeed, the butterfly now does better at these sites. Now, if you run a metapopulation modeling network where you improve the quality of these previously bad sites, that of course will improve the demographic performance of the metapopulation, which is shown by the simulation here. And in this first example, we don't allow any evolutionary shift in host plant preference of the butterflies. It still prefers the host plant on the warm sites, doesn't prefer the host species on the cool sites. So it can develop on that, but not as well as on the plant on the warm sites. But if you now turn on the evolution, allow the species also to evolve in terms of its host plant preference, we observe an evolution towards a generalist species, which now kind of makes a compromise between being well adapted to the warm side plant or the cool side plant. Now it does reasonably well in both cases, and that actually will increase the metapopulation size, which is consistent with what is empirically known for the species. All right, now I next move on to talk about my own butterfly species, and I do this in the context of a kind of variant of the model where we describe the dynamics of the metapopulation in terms of the probability of patches in a network being occupied and the mean phenotype in occupied patches. In this example, the mean phenotype now is the frequency of fast dispersing individuals in the local population. So there are two kinds of individuals, those which are fast dispersing, those which are slow dispersing, and our mean phenotype now is the frequency of the fast dispersing individual in a particular population. Again, this work has been recently published, so there's no need to go into details. I just emphasize that in the model one variable which has an importance is the mean phenotype of the immigrants. So a few slides about the butterfly system which I've been studying for the past 20 years on a large island in southwestern Finland. We have mapped on this very large scale of 50 by 70 kilometers a network of little meadows, about 4,000 of them. We keep on doing a long-term study where we survey every year the presence or absence of the butterfly in the meadows, and of course we do lots of other things as well. So this map here shows the pattern of occupancy in this network. The black dots are occupied meadows in one particular year. Now, we have done a lot of work on dispersal for obvious reasons of the butterfly, including an experiment which we did some years ago with people from Rothamsted Research in the UK using an equipment called harmonic radar to track free-flying butterflies. And for that purpose, we attached a little, it's not so little, but it's a very light transponder with an antenna on a butterfly, which didn't really seem to affect its behavior. And then we tracked the movement of these butterflies with a little antenna on their back on this flat heathland in East Anglia. Amazing technology. You can actually follow a butterfly up to 1 kilometer with a spatial resolution of only a couple of meters. So one result that came out of that exercise was, we reared a set of butterflies for the experiment and transported them from Finland to England. Well, it was a very special set of butterflies. There was variation in a polymorphic gene called phosphoglucose isomerase, which very many of you will recognize as the classic enzyme which Ward Watt did in Stanford did pioneering work using another butterfly, Goliath's butterfly 35 years ago. Well, the same enzyme, same gene is also polymorphic in our butterfly, and we have identified a single nucleotide polymorphism, a SNP, in the coding region of that gene, which is very strongly associated with a number of life history traits, including now dispersal rate in the field as measured by the harmonic radar. So here I'm comparing individuals which are either heterozygous for that SNP or homozygous for the major allele. On the y-axis we have the distance, log distance moved in 5 minutes based on the radar tracking. Well, we track the butterflies actually for an entire day, but this is the average distance they moved in 5 minutes against ambient air temperature, which also has an influence. These are not the actual measured data points. These are the fitted lines to the empirical data. There was some scatter there, but as you can see, there was a big systematic difference and significant difference between the two genotypes. So interestingly also a significant interaction with the ambient temperature. Now here in California, I guess the two butterfly genotypes would do equally well because the temperatures, ambient temperatures are more like here. In our climate they are more like here, and there is a big difference between the dispersal distances of the two genotypes, which can be demonstrated very simply by collecting individuals from newly established populations and old populations and comparing the genotypic composition, which we have done here. Of course, you would expect that if one genotype is much more dispersive, then these are the ones which mostly establish new populations, and their offspring also tend to have lots of same genotype, or in this case, the heterozygous form. Now, you might think that this is a rather small data set to demonstrate that. It is kind of small, but on the other hand, it's very high quality because each individual was collected from a different local population. So it was sort of maximally replicated and a highly significant difference in the expected direction. So there is a difference between newly established and old populations in the PGI genotype for obvious reason. There is also an interesting pattern at a larger scale. This map shows the mean phenotype in the local populations in terms of the frequency of the more dispersive AC heterozygous individuals. Darker is the color, greater is the frequency of the more dispersive individual. And we can see that there's a very strong spatially correlated pattern such that there are regions, regardless of whether the population is old or new or what is the age, the frequency of the more dispersive individuals is high, and there are regions where it is low. So now going back to the model, we can use the model, or we can in principle anyway, we can fit the model to this empirical data of this type. And for that purpose, you first solve the equilibrium frequency of fast dispersing individuals in these n populations in your network, which is done here. So Q sub I is the frequency of the fast dispersing individuals at equilibrium in the populations. Now the explanatory variables in the model are colonization rate, extinction rate, and the frequency of fast dispersing individuals among the immigrants that are coming to the population. Now these can't be measured empirically. You can't really measure the genotype or the phenotype of every immigrant to every population because you can't catch them all. But you can use proxies, or we can use proxies which can be measured empirically, which are well justified biologically as measures, proxy measures of the colonization rate, extinction rate, and the frequency of fast dispersing individuals among the immigrants. Now, one problem with this model is that it's rather complex, and it actually fits the data pretty well. It explains about 33% of the variation in the frequency of fast dispersing individuals among these many local populations. But I'm kind of worried that it's too complex, and of course the structure is not exactly correct, so it's not a very robust model perhaps. And also when you look at the model more closely, you actually realize that the linear effects of colonization rate, extinction rate, and the frequency of fast dispersing individuals might, they really should be the dominant effects. And therefore, we just constructed a very simple linear multiple regression model where we have now these explanatory variables. We are explaining simply the spatial variation with this linear model in the frequency of these fast dispersing individuals. And indeed, it does actually a little better than the correct nonlinear model. And all these factors, they have a significant effect. So higher is the extinction rate, higher is the proxy of the extinction rate, higher is the frequency of fast dispersing individuals in that population. And why is that? It is because if a particular population in the long course of time has high extinction rate, there is also a high rate of establishment, there is high founder effect, and if the founders typically are fast dispersing, then you would expect in that population higher frequency of the fast dispersing in the long course of time. If a particular population has a high colonization rate, same thing, high frequency of fast dispersing individuals. Also, if the frequency of the fast dispersing individuals is high among the immigrants, that will pump up the frequency of fast dispersing individuals in that population. And also, I actually added here the age of the population, even though it doesn't really appear in this deterministic approximation, which is only for the long-term frequency of the mob. But in this linear model, you can actually include that. And also, it made a significant contribution. And altogether, this model explains more than 40% of the variation in the frequency of this fast dispersing genotype among the population, which I find really quite amazing that you can explain with these factors so much of the spatial variation in the genotypic frequency. Also, the model predicts this spatial autocorrelation in the gene frequency. It predicts it because the range of dispersal of the butterflies is rather short. That leads to a spatially correlated pattern. If the range of dispersal would be larger, then of course that spatially correlated pattern would disappear. All right, I'm keeping my time. I think I came to my last slide. Some final thoughts about what we could, what I anyway call eco-evolutionary spatial dynamics. I guess one could expect that they are particularly important when there is some change in the environmental conditions. Like in the brown Argus butterfly example, there is climate change induced evolution of dispersal rate, possibly, but definitely there has been evolution of the host plant preference that has influenced the range expansion, which for the brown Argus there is no evidence on this, but you could well expect that would reinforce selection on dispersal. So very sort of clear example of a situation where the demographic dynamics and the evolutionary dynamics might be expected to be coupled. In classic metapopulation, there is no sort of systematic range expansion, but because of this continuous turnover of populations, the demographic composition of populations is all the time changing. So from the point of view of individuals, conditions are changing in time. And then there are examples, a really nice paper by Wasser et al. In AMNAT last year, where they showed that the sort of very small-scale spatial variation in a plant community, which would be due to the fact that a particular plant individual may be either surrounded by conspecifics or heterospecifics. Now, if that would affect the selection on the focal individual, that pattern of whether you are surrounded by conspecifics or heterospecifics is changing all the time. And again, that is a situation where one could expect coupling between the demographic and microevolutionary dynamics. Now, this sort of coupling oftentimes could be expected to be a mechanism, perhaps even an important mechanism that maintains genetic and life history variation. And as my example showed, it may also either increase or decrease metapopulation size in, let's say, deteriorating environments where there might be habitat loss and fragmentation. On the other hand, I think very generally it is likely to be the case that in a deteriorating environment there may well be some evolutionary response which kind of improves the performance, the demographic performance of the population, but it is oftentimes unlikely that this improvement due to this micro-evolutionary change would be so strong that it would kind of completely compensate the direct ecological harm that the deteriorating environment is doing. Like in the case of habitat loss and fragmentation, there is a direct ecological harm done to the population. Now there may be some adjustment, say in the dispersal rate, which may improve the chances of the population persisting in spite of habitat loss and fragmentation, but that adjustment very unlikely would be so strong that it would completely compensate the direct ecological response. Thanks very much.
Sure, Ilkka would be
Sure.
happy to answer questions.
Sorry, but I probably had to rush a bit too fast. I realized in the middle that I had too many slides. Yeah.
So, going back to the metapopulations, is the effect that you're finding of those four kinds of adaptation, how does that relate to these sort of ideas that maybe, I guess, Holt and Galbakaiewicz looked at where things get swamped and there's too much dispersal, you can't adapt, etc. Is it really sort of the same thing but expanded to more than two?
Well, absolutely. I mean, this case of habitat generalization happens because of a lot of gene flow, or that is one factor which tends to push the spatial adaptation in that direction. Of course, it also depends on the other model parameters, like if you have weak selection, then that also is likely to lead to such a generalist type of adaptation. Or, as I mentioned, if the difference between the optimal phenotypes between the two habitat types is small, then again it sort of doesn't pay to specialize. So it certainly is here, and I don't think that going to a network from the situation where you say I only have two populations would lead to dramatically different predictions. This mosaic specialization, of course, is a kind of pattern which you can't see if you only have two patches, but it's sort of understandable. But what I'm sort of interested about these types of models is that you know, they reflect, in my mind, pretty well the actual reality in many fragmented landscapes, and you can actually parameterize, or you can try to parameterize them with empirical data, like we had done with the walking stick insect. And to me, that's a kind of step forward in comparison with models which I think are very valuable, and the, you know, the conceptual insights are more or less the same, but which you still can't relate so closely with empirical data. Yes. And
so, as you know, I'm interested in this phenomenon of phenotype-dependent dispersal. So the dispersers not just being more dispersive than the others, but carrying with them an entire suite of traits that could be that it's just physiologically more geared up so they can fly better. And I know we were talking about that earlier, but it could also be things that make them good at settling in some habitats where you'd expect them to be good at habitat selection, or good at even cognitively, you know, being plastic, because they're going to end up in new places all the time. What sorts of things, I know you know a lot about that kind of thing. Could you just sort of elaborate some about what you know about that?
well, I mean, in this particular case, it's pretty obvious that the more dispersive type, if we now relate it back to the genetic polymorphism in this gene, phosphoglucose isomerase, does carry this trait, this suite of other traits with it, because we actually know empirically that the same polymorphism is affecting many other life history traits. Yeah. Which is, by the way, it's kind of surprising that there is this one single actually nucleotide which has all these amazing associations about which I didn't have time to talk about, but which are replicated, which really are true effects. Now this is a glycolytic, this is a gene which is encoding an enzyme in the glycolytic pathway, and the old idea has been, starting from Watt, that you know, the different isoforms of the enzyme, they have different kinetic properties, and they are catalyzing this you know, sugar metabolism in different way, and you know, the interaction with temperature is consistent with that idea. Now, for the past 5 years, I've been trying, in collaboration with people in Helsinki, to develop this story to the level where we actually would be able to complement these associations with a proper molecular level understanding of what actually is going on. But we have failed, and we now think that perhaps this glycolytic effect of this enzyme is not at least the full story. It is known that PGI molecule actually is also has the other function, being a signaling molecule in many other pathways apart from the glycolytic pathway. And it may actually be that this signaling function of this same molecule is somehow underlying these associations, which would be equally fascinating story, but unfortunately we have not really been able to make much progress with that. But it's a very special gene. Yes.
The walking stick example, is there an effect of the quality of the different host plants for the different genotypes that's built into the model, if there is any such?
No. The model, of course, is very simple, and you know, there's just this one parameter which is determining the strength of local selection. And my understanding is really that this bird predation is the selective factor. So, you know, I wouldn't be surprised if there would be something else going on here, and there probably are people in the audience who know so much more about the walking stick insect and its host plants than I do. But this bird predation is certainly is one factor which has been known for a long time. So I guess this is the first time that it has been demonstrated that this local adaptation actually does happen at the push level. You know, the previous work has been done at the somewhat larger spatial scale. Yes?
I'm interested to hear your thoughts on the trend on the genetic side towards collecting more and more and more markers and genotype by sequencing. And in your field, do you feel like there's still questions that can be answered with microsatellite analysis or smaller numbers of SNPs?
Well, my answer is that I have spent a huge amount of money, much more than you can imagine, on sequencing the full genome over the past 3 years of our butterfly, and we are now hopefully coming to the end, towards the end of this summer. So I guess I must say that I do have some trust on some good things coming out of knowing much more about the genome-wide molecular variation in the species. So, you know, at the same time, you know, I do realize that it may be a bit naive to just assume that you get, you know, a fantastic insight through this sort of associational linkage analysis. So, you know, there are likely to be other genes which are polymorphic, variable, which also are associated, like PGI, with some life history traits. But exactly how one should go about explaining phenotypic or life history variation with genetic variation, if one is interested in figuring out the genetic basis of phenotypic life history variation, it's another matter. There's one really interesting piece of work that has been done over the past few years, and some of you may know about this well, but I myself just came to know about it recently, and I can explain this with the example of human height, which has very high heritability, and people have been trying to find the genetic basis of the human height for a long time, trying to find SNPs that would explain large part of that heritability, and by and large this has failed. It has failed because even though you score 10,000 SNPs, you don't find small number of SNPs that would be very strongly associated with height. However, recently, within the past few years, people have taken a sort of to me, very sensible and simple approach where you don't kind of ignore the markers, the SNPs, which are not significantly associated with height in this example. But you actually construct a model where you include also the non-significant effects. So you may have a model which includes 2,000 SNPs, and you take at the face value the strength of association with all these markers, which on their own are not significant, not particularly when you correct for multiple testing. And quite amazingly, that kind of approach gives you a highly predictive model. So the message here is that there is not a single genetic variant that would explain this particular quantitative trait well. There are many, many genes which do contribute, but actually you can, if you have lots of information for thousands of genes, you can actually construct a predictive model in a very simple and straightforward manner. So PGI probably is a very, very exceptional gene in terms of having this strong single gene or even single nucleotide effect. So of course, we don't know whether the SNP that we have been using, whether it's functionally the important variant, but it's close to some functionally important variation. But the hope is that if you have enough genome-wide knowledge of genetic variation, you might be able to build up understanding also of traits which are affected by many genes. Sorry about rather long answer.